Can you guys? Oh, all right. So I'm as loud as I'm going to get. Can everyone hear me fairly well? Okay. All right. So I sort of ran ahead of myself with the title of this talk. So I'm probably not going to get to all of it, but hopefully this is going to be somehow interesting. So if I am completely out of the comfort zone here, let me know. Stop me mid-talk so I can correct my course. I do talk mostly to biologists, so if this is not good for you, let me know. Okay, so one of the first things that I found that when I started working on this problem is that there are really cool pictures out there. So people make an effort to visualize the interaction of life on Earth with geological record really well. They make an effort, really make an effort. This is one of them. So basically, you could see the evolutionary period together with the dates or eras listed on it. The other way to look at it is perhaps from the biological perspective is this story of life. We have this tree of life, and some people debate whether it's a tree or a bush, and I tend to lean towards the latter. But what we find is that supposedly we had some origin of life, and that everything else had evolved from that particular origin. And the way that people look at it in the biological world is by taking count of how many mutations in the DNA record have occurred, and 2 organisms are said to be diversified by the number of these mutations, right? So the less mutations you've had, supposedly the less time had passed, and so therefore you can say that these 2 organisms are closer related. So here it's a nice visual representation. We usually aren't that great in making these representations, but the tree here, this is millions of years. So this is the root, and then organisms start differentiating. So you go from bacteria, archaea, eukaryotes, and so on and so forth. And this is obviously focused on eukaryotes, and actually in the real world, bacteria rule. There are significantly more bacteria than there are eukaryotes, not just by cell count, but also by the number of types, right? So there are different types. So how do people look at this evolution? Well, as I said before, imagine that this is the DNA sequence of species A, species B, species C, and so on and so forth. And the longer it takes for the species, the further the species are separated, the more so-called mutations or differences in the genetic code highlighted by these bold letters over here in the alignment you will see collected. So then you can compute the distance between every species on the basis of this multiple sequence alignment in the number of the letters in the DNA that is different between them. You need the multiple sequence alignment in order to be able to trace the history across all species, but at any given point in time, you only look at 2 species in particular, and you define the distance between them. And then you can build this tree. This is a hierarchical structure here, on the basis of who is closest together. And there are many ways of building the tree. So the catch with the genome is that usually when you look at the differences and similarities of the genomes, you are actually interested in the functional components of the genome. And in bacteria, this is roughly 80 to 90% of the genome. The genes make up roughly 80 to 90%. In eukaryotes, it's significantly less. But we argue that you can trace record most in the functional components, so in the genes. Now, genes encode proteins, as I'm sure all of you know. And in proteins, things get a little more complicated. You have a protein sequence, which is directly translated from the RNA sequence, which is copied from the DNA. This primary sequence of the protein folds into, or often folds into secondary structures. With the exception of the disordered proteins, this is the way that most proteins go. Secondary structures form tertiary structures and form quaternary structures by interaction. So basically, it's also a hierarchical folding model, right? So a native state or a native conformation is where the protein lives most of its time and where it performs most of its activity, okay? So this is a figure from Ford Doolittle's publication where he said, well, you know what? The sequence information that we can capture from genes, from genetic alignments, the stuff that I showed you before, is actually fairly limited. So this is approximation, right? But at about 20% sequence identity, you can still do multiple sequence alignments in order to capture the evolutionary process of the organisms across the genes that you're interested in. But around 10%, and if you're going back in deep time to the origins of life, you're probably not going to get 20% sequence identity across genes. Some of the genes have not existed that many years ago, and they would have to arise independently or from other genes. So the sequence identity between genes is actually quite limited. The further down you go, the further away you go. But if you look at the 3D structure of the proteins, because think about this, proteins are the molecular functionality. These are the things that organisms need to perform in order to survive, right? So they need to catalyze certain reactions. They need to, whatever an organism needs to do, it will accomplish via, or most of the things, it will accomplish via its proteins. So when we say that evolution or selection acts on organisms, what we mean is we are selecting for organisms that can encode proteins that are most functional in a particular environment. So while looking at genetic similarity, at sequence similarity, we can talk about distance and years in terms of accumulation of mutations. If we look at protein structure and hereby protein function, we can actually figure out whether organisms are related functionally. Okay, even if we don't see sequence similarity. So this is what protein structures look like. This is protein 1, protein 2. This thing is called the beta sheet. This is the basic idea of secondary structure. This is what the basic secondary structure components may look like. So if I asked you to align these two proteins, well, if I ask you to align anything, well, it's really quite simple. You just put it on top of another, and it's aligned. Right? There is nothing else to be going on there, except well, this is what this alignment looks like. Now, can someone tell me if this is actually a good alignment or a bad alignment? Well, that's the thing. I don't know. It looks good, right? But sometimes, it's kind of like that, right? So the idea is that you can align an apple on an orange. You can superimpose them. Doesn't mean that one does what the other is intended to do. So, and in proteins, it's actually quite complicated. This is NMR structures of the same protein with 3 residues, 3 amino acids substituted. And they're indicated in this red. And the reason why it looks like there's many of them is because it's an NMR structure, so it kind of captures the probabilistic display of the protein, right? And what you see here is there's a helix here, there's a helix here. You have 3 mutations, these 3 residues, right? And all of a sudden, the helices are converted to beta sheets, right? And in biological terms, this is most often not going to do what you want it to do. So the moment you've had that mutation, this protein is gone. And that's only 3 mutations, right? So it's not a very far stretch in biological terms. So how do we know whether a particular structural similarity actually has any meaning to it, right? So the first thing is, this is what a big, huge protein looks like. If we try to look over the entire protein, we're probably going to get a lot of this accessory information, which is probably not that interesting in large length or spans of time. So what we want to look at is this active site, or the most important functional site of the protein. And in our case, we were interested in metal binding, right, in metal ion binding. And we actually found that if we focus, if we superimpose a sphere, just sort of extract every single amino acid within a radius of 15 angstroms from the center of the metal ligand that we have, here it's an iron-sulfur cluster, then we would be able to extract the functional side of the protein, right? You can think of it as the car engine, whereas the car can change, the car engine kind of tends to stay the same, right? At least for now. Okay, so here are 2 more alignments, right? And this is the way to look at them is there are 2 proteins, one in blue and one in green. And when they overlap, one color becomes orange and one color becomes red, right? So what you want to do for a good alignment is look for orange and red in this particular scenario. So when you look at these two alignments, you kind of see that this is a better alignment than that is, right? But overall, these pieces align better here than there. So if you actually looked at the generic metrics of how we measure whether alignment is correct or not, and this is done via root mean square distance between the individual atoms of the protein structures, right? Here, over the entire range, we get a distance of 1.91. And here, you get a distance of 1.04. Okay? And if we use the standard metrics, this would be a better alignment than this. But just intuitively, when you compare two protein structures and you're looking for functional similarity, it's actually quite clear that this one is probably a better match, right? And what we suspected is that actually, if we looked at the center distances, so here it's very visible, the distance between the centers of the ligands, right? We could actually get a better understanding of whether the superimposition of the protein structures is meaningful or not. So because you need to bind this particular ligand in order to perform the functionality that you are meant to perform, then you need to do this in a similar way in order to claim the same functionality. So here, the Euclidean distance is 19 angstroms. This is the distance between the centers. Here, it's less than 1 angstrom, so this is probably a better bet. So we actually asked this question for alignments of protein structures. This is all protein structures that we can get where we're aligning iron-sulfur clusters and iron-sulfur cluster and heme-binding proteins, right? So we're aligning their centers, their 15 angstrom spheres. And what we see is that the x-axis here is the cofactor distance, and we see this distribution in terms of the number of alignments of the ligand distances, right? So these distances between the ligands that the proteins bind. So it looks like there's very few over here in the small distance, which is what we want, right? And actually, this is what you expect. Most of the proteins carry out different functions. So if you align them, their cofactors or their ligands should not be close together, right? On the other hand, if we took the proteins of high similarity, so the ones that high functional similarity here, we would see that there is this peak at the 1, 2, 3 cofactor distance, right? And we could then actually quantify being able to see functional similarity using cofactor distances, right? So that was something that we found that was quite interesting. And we saw that for heme-binding proteins, if the cofactors were less than 3 angstroms away, we could have up to 90% accuracy in identifying proteins that perform the same function versus identifying proteins that perform different functions. And this is not trivial in protein structure world. Although people believe that determining a protein structure is enough to identify its molecular functionality, this is not the case. And so we were very excited about this particular finding. For this particular iron-sulfur cluster, we needed less than 2.8 angstroms, and for this one, we needed less than 2.1 angstrom distance in order to be correct. So we said that actually we want to be conservative, and for any one of them, anything less than 2 angstroms was going to be considered a correct alignment. Now, maybe this was an overly conservative estimate, but less than 2 angstroms is what we decided was going to be the way for us to identify whether two particular proteins with a given structure actually perform the same function. So the catch there was that all of these proteins had bound the same ligand, right? So they, like here, you see there's 2 proteins. They're binding a heme. So remember, the red and the orange means the proteins overlap. These proteins overlap really, really well. And they're binding 2 different hemes, and that's why it's in the colors of Ukrainian flag, right? In the blue and yellow here. But the reality is we're not really always interested in heme-binding things. We could also be interested in things that bind other ligands. In this case, is a cobalt ion over here, right? So this alignment of protein structure still looks really good, right? But they're binding the very different ions, right? Very different ligands in this case. And so how do we know whether these two proteins perform the same function? Okay. So one thing that we wanted to put back into the equation was the story of sequence alignment. Remember, sequence alignments actually give us some information about how far apart two sequences are in time. You need more time to accumulate more mutations. Okay? So the idea is if we take this protein structure, we can actually say what is the sequence alignment from the protein structure that we can extract, right? So this is a residue in this amino acid in this orange and amino acid in the red, and they're next to each other, and we call that an alignment, okay? So these structures, as you can tell, they're disjoint. They're not following each other. So it's not sequence in terms of sequence of letters. It's sequence in terms of just the structure here, right? So we asked this question. For looking at the structure, can we identify some sequence properties that would tell us whether two genes, or two proteins in this case already, actually have the same functionality? And what we could do is we could count the number of amino acids that have aligned, and in this case is 55. So the alignment length here is 55 amino acids. And then we can ask the question of how many of these amino acids are identical, right? So proline here, an aspartic acid, a glutamic acid, and so on and so forth. And actually, it's 14 out of 55, and that's 25% sequence identity. And if you recall, you need 30% sequence identity to somehow transfer similarity. And what is 25%? And what we decided to do in this particular case is to try to figure out if there is something different about the proteins that are of the same functionality versus proteins of the different functionality in terms of the sequence identity and alignment length, those two parameters that I had told you about. And how do I know if something is of the same functionality or not? Well, long ligand distance. Remember that 2 angstrom cutoff? Well, if it was above 2 angstroms, it's different functionality, right? So if I overlap two structures, if it's above 2 angstrom, it's different functionality. And I can map onto this graph the alignments of structures. Each point represents an alignment of two structures with their alignment length and sequence identity, okay? So these guys are the ones you don't want. Now, this is what the guys that you do want look like, right? So this is the ones that have short ligand distance. Okay, so what I can do is then, you could see that these are quite different, is you can superimpose them and try to optimize a curve, and this is just a gradient decay, an exponential gradient decay curve here, which is not perfect, but it separates the alignments of structures which have the same functionality from the ones that have a different functionality. And now I no longer need a ligand. All I need is a structural alignment. Is that clear? Cool, because this is like the key, the key point here. So the idea is that if you go away from the curve that I had just shown you, go up in sequence identity, you get a lot more reliable predictions. And the further up you go, the better you get. So 0 is the curve, right? And so if I go 5% sequence identity more up from the curve, right, that's going to give me higher precision, higher accuracy in naming things at the cost of the recall of how many of the pairs that do perform the same function do I actually get. And so the idea here is very simple. This is the best balance. That's how we optimize the curve, to have the best balance between the recall and the precision. But if I wanted to be 100% sure in everything that I called, I would probably just go left here. Just pick a different threshold for my cutoff. Okay, so this is cool. This also means that the sahle threshold, and we call this sahle, this scar is called sahle, don't structure alignment, informed homology, something, but whatever, it's sahle. And it can be used to evaluate different ligand-protein structure alignments. Right, so in this past, 15 minutes or so, I just told you that I can solve a problem that people have been trying to solve for many years. don't believe me, but we can approximate. Okay. So now I can take all the protein structures that I want, and I can make alignments with them. And I was interested in the evolution of biological redox. And for that, I needed all proteins that bind transition metals. And I excluded zinc from this consideration because zinc is very often used for transcription activities, for DNA regulation activities, and I didn't want them in there. I was interested in redox per se. And I also excluded the mercury from this consideration for purely experimental bias reasons, because mercury is most often used to fill in whatever it is that you want to fill in, whatever metal you want to fill in order to be able to determine the crystal structure of the protein. Okay? So what we have here is the iron ligands, manganese ligands, copper ligands, and so on and so forth. And we extracted 4,634 spheres altogether from the protein data bank, which is, by the way, upwards of 120,000 structures. So this is quite a small number. And the reason the number is so small is because most people determine structures of already known structural proteins, right? So you will go out and you will mutate the protein and see what effect a particular mutation has on protein structure, and that will be a new structure that you deposit into the protein data bank. So if you wanted things that were wild type, that were non-redundant in terms of their sequences, but very interesting in terms of their structures, very diverse in terms of their structures, you would go here. It's really not a very large set. I mean, there are 80 million proteins that we think we have right now, sequenced right now. So 4,000 spheres is not too much. But we believe that it is very representative of the reality of biological redox of the world. Okay, and then we can put edges onto this graph from the alignments of these structures. And we can compute the sahle scores for each one of these alignments. And if it's sahle less than 0, then we're not interested. Okay? So that's not an edge that we want. This is what the network looks like. So this is the distribution of the metals in this particular network. You can see that the majority of the ligands here are iron-based. Some manganese, some copper, cobalt, and so on and so forth. The interesting architecture of this network is somehow alluring to start making conclusions, right? So this is, thanks Bob for pointing this out, but this is the iron, the manganese to copper timeline in the network. Well, and by the way, the largest connected component of this structure network contains 94% of those 4,600 spheres, which in itself is telling. It means that we can find functional similarity across all of these proteins, and sort of begs this idea that maybe they come from the same ancestor. Maybe. So just as a sidetrack here, and just to make sure that we're on the same page, there's such a notion as convergent evolution. And convergent evolution basically means you're trying to do the same thing from different ends. So you evolve the same functionality twice. In sequence land, that almost never happens, because if you have the same sequence, it's very unlikely that you at random create that same sequence twice. But it is possible in structure land. Right? So you can create the same structures that are active. Here, the sahle score contains sequence information. So that may or may not be convergent, and we have yet to think about this at length and in detail. But so now this is iron, manganese, and copper, but then you think about the forms that iron comes in, and you see that actually, well, this is heme, this is the iron ions, this is the iron sulfur clusters over here, and there's more iron there. And so while the manganese to copper timeline is probably still there, instead of the iron to manganese, it could also be that, or it could be, it could be that. Right? So the problem, the biggest problem that we have here is that we have no reference. So we have a structural alignment that is somehow sequence-based because of the nature of the sahle score, but really we don't know anything in terms of timing these things or even figuring out how the relationship is likely to have formed, okay, to begin with. So what we wanted to do is we wanted to see if we can somehow anchor the predictions that we were making or the annotations that we were making in other stuff, so in other people's work. In this particular case, we collected this tree that was conveniently created by Jonathan Eisen's group in 2013. This is bacteria and archaea. This is a really large tree of I think, something like 1,200 organisms. And what he did is he extracted 24 genes from every single one of these organisms. And these were the ribosomal genes, which are super important for activity of any organism, of the life of every organism. And he aligned them. He did the sequence alignment. So basically, this tree is actually an evolutionary tree of organisms on the basis of these 24 genes that have nothing to do with anything that I'm interested in, right? But these are bacterial arrangements, and even though I can't really put a date on any one of these splits, I can tell you how far away from the root is any one of these particular organisms. So how far away from the ancestor, so how like the ancestor it is, and also what is the distance in evolutionary terms between any two organisms in this tree, right? Okay, so what does that help me? Well, I can actually take the protein sequences that determine the structures that I've been talking about up until now and align them to the genomes, the complete genomes of these microorganisms. Let me repeat myself one more time. This tree is built on 24 genes selected from every single genome of these microorganisms. These genes are related to transcription translation machinery of the microorganism, have nothing to do with redox, right? So this is an independent tree. Now, what I'm doing is I'm asking if I take my redox proteins and I align them to the genomes, can I find the matches of these particular proteins? And it turns out I can. So in 3,000 cases of the 4,354 proteins in our network, we can map them to an organism. And we can actually figure out that 42% of our edges in the network correlate to the distance between the organisms. And so then we asked, well, now is there a correlation between our scores, which have nothing to do with this particular world, and the ages or the distances between any two organisms in this tree? And the correlation is quite high. And it's negative because the higher the sahle score, the shorter the distance between them, right? So the more like them. Pretty cool. I was very happy about this. But then I said, OK, what if I exclude zero distance hits, which basically means this protein or some ancestor of this protein exists in this particular organism, right? So these two proteins are in the same organism. And that happens quite a lot, apparently, which says one of two things. Either we need to go back and up our resolution in terms of mapping proteins to organisms, actually loosen our resolution because we are being very strict about this and maybe we need to go looser, or that there is a lot of duplication going on in ancestors giving rise to all of these proteins that we now observe, which is a very intriguing finding. So either way, whether the correlation is there, which is great, yay, we have an evolutionary way of tracing, evolution-based way of tracing evolution of redox, biological redox, or we have a single ancestor or very few ancestors that produce this diversity that we're observing now, which is very cool. So the other thing that we wanted to check, which was actually a failed attempt by me on an earlier time, so it took me a year to figure this out, but one of the most interesting things here is that you can compute the distance between any two proteins using the edges. So this is the shortest path between 1 and 2, but really you could have another path and another path and so on and so forth. So what you want is you compute the shortest path because that's the most likely situation in evolution. And you can do that for every single protein in the group, and you can compute that matrix, and then you can ask, is the shortest path distance correlated with evolutionary distance? And actually, it's not. So on the one hand, I was disappointed because I had all of these ideas that I could test with this particular situation. On the other hand, I was actually quite excited seeing that my sahle score is actually representative of evolution non-randomly, because this is basically a random correlation. So there was one more thing. Yeah? The map between proteins and... Right, exactly. So I say if the protein homologue, the protein similar protein, exists in a bacterial genome, I take the oldest organism where I can find that homologue, and I assign that organism to this bug, to this protein. And then the distance between two organisms is the distance that I'm correlating to the sahle score. Does that make sense? Okay. Yeah? I'm sorry, horizontal gene transfer? Yeah, so that's the thing. I'm not taking horizontal gene transfer into account, and that was the reason why I said, well, maybe this, if you remove the single organism maps, hits, then you get a lower correlation. So that to me means that there was a single ancestor. If that's true, that was a single ancestor that was horizontally transferred all over the world. So I'm not saying it outright yet because I have to check every other vertical descent question before I say that, but that seems to be the case. Yeah? For the shortest path, it doesn't matter in this case. We did a couple of implementations. Shortest path don't give us a correlation. I don't remember which one I used for this particular correlation, but we used a couple of algorithms for depth first and breadth first, and there is approximation algorithms as well. Anything else? Okay. So the problem that we had to deal with, another problem that we had to deal with is the experimental bias in the PDB, in the protein data bank. And the reason, as I told you, is because people want to determine structures of mutants of the same protein 2,500 times. So actually, I was very surprised. The PDB is at Rutgers, where I am, and I talked to Helen Berman, who was the head of the PDB for the longest time. And she told me that in the 80s, 25% of PDB was hemoglobin. So I found that quite interesting. Why would you determine the structure of the same protein all the time? Well, the answer is because you could. Right? And that was the idea. So what we did is we tried to eliminate the experimental bias. The way to do it, we said we're going to extract the metal or ligand-based subnetworks, right? So if you remember this was our original network. We only took, let's say, this side. This is the iron subnetwork. And then we clustered the iron subnetwork into individual clusters. And for every cluster, we said, okay, we're going to take the center of every cluster. And I'm going to spare you the details, but if you're interested, I can actually talk about the C index and the clustering of this thing. But we took the center of these clusters, and we said, now what we need to do, we had 311 nodes, which represented the reduced set, but very representative of the entire network. So what we wanted to do now is find a Steiner tree. A Steiner tree is basically if you have a network and the red nodes are the ones that you want to cover, right? The Steiner tree is the minimal tree across this network that will cover those nodes, okay? So what we wanted to do is find a Steiner tree. Steiner tree is an extremely hard problem, so we did an approximation of the Steiner tree, and we were able to recall 283 of 311 representatives, so these 311, and we were able to cover them in 506 nodes. So now of our 4,300 nodes, we're at the tree of roughly 500 nodes, And excuse me for the switch of colors, but I wasn't, in the time that I had, wasn't able to reproduce the pie chart. Didn't realize that this was going to happen. But you see that the distribution of the individual metals is actually very different from the distribution here. Right? So, yeah. So the catch was that we wanted to make sure that this correlation still holds for this particular reduced set, and it sort of did. I mean, it did lose some accuracy here, but this is what the network looks like now, and all of a sudden it's no longer ligand specific, right? So this is the network with the intermediate steps, right? So these connectors, not just the terminals. And this is the network with just the terminals. And you could see that all the ligands are sort of mixed. Right? So actually, I'm very excited about this. This, to me, tells me that there was some sort of small number subset of origins, and they have diversified into the entire variety we're observing. The fact that the network is still connected at this stage is very telling. Yes, I'm forcing it to be connected, but the fact that I can force it to be connected is very telling to me of the fact that it could have been single, or not single origin, but very small number of origins of this particular activity. Here, I applied a PageRank algorithm, basically trying to figure out if I start from any node in the network, how likely am I to hit a particular node. So these nodes that you see highlighted in larger size are the ones that show up. Now, this is a work in progress, so there are many algorithms that I can apply here. Centrality, for instance, looking for centrality would be one. I just thought that PageRank would be an interesting one. It's just using basic Google base. So this is Sergey Brin's advance in Google many years back. So I thought this would be fun to apply here. But these guys show up, right? And actually, if you look at them, they make sense. So this one, there's 4 that I showed you. This is a manganese center of Bacillus subtilis oxalate decarboxylase, right? So, and the structure was determined with cobalt. It's a manganese binding structure that was done with cobalt, suggesting already that these ions are replaceable for this particular activity. This guy, by the way, is mostly an Arab, but it has a very interesting lifestyle when you force it to be an anaerobe, which is also something that I was looking for, thinking that actually the first proteins would have to be applicable in anaerobic conditions. This guy is an obligate anaerobe, so this is a bacterioferritin from this Bacteroides. And, well, this one is unfortunately aerobe, right? So, but I guess that's the beginning of, that's the end of some evolutionary line. And this one is also sometimes anaerobe, sometimes aerobe. So 3 of the 4 are actually currently in organisms which could very well grow in anaerobic conditions. It's very reassuring in this particular case. You cannot see connections for some reason. I don't know. Sorry about that, but that's a tree. So if I took Eisen's tree, the tree that I showed you of the organisms, and just extracted the piece that was relevant to us, and I highlighted our structures. So this is the center. I'm sorry that you can't see the connections. This is the center. Anything that's closest to the center is the more ancestral-like protein. So if you look at these guys who are closest, this is the one, this is the ancient one, which potentially is due to horizontal gene transfer, diversification of a particular protein functionality. But these guys have some, I guess, so if you see a line like this, a straight line, it means it's one organism that's mapping to it. So if you have multiple proteins that map to a single organism, that suggests that either that is the closest to that is probably the closest to the ancestral gene that you have. So if you look at the tree with just the terminals, you also see similarity, similar thing. You see no copper. Copper is orange, and there is no copper that appears very close, but that appears fairly fast after, suggesting not that copper was available for activity at that point, but that the copper binding protein would have come from that ancestor as well. Okay, so one final slide I think that I want to show, if I haven't lost all of you yet, is this network, back to this network that we had. These are the oldest proteins in the mix. The green means they're younger. The small blue are older, right? So what you're seeing is that same network that I showed you before with all the proteins being distributed across the entire network. And you see that in almost every cluster, you get an origin, which is old, right? And that, to me, is also reassuring, suggesting that either these things have duplicated in the same organism and then have diverged to give everything else, or they have converged to the functionality which was necessary to everybody else. Okay, so in conclusion, there was no explicit input of the time variable in any step of the analysis. I never put it in, right, until the correlation came about. And yet, we see time, right? It correlates. We need to resolve the mapping to the same organism by multiple proteins. That is a critical question. We need to understand why it's happening. And my guess is horizontal gene transfer. It appears that a large fraction of metal ligand binding mediated activity has evolved on the basis of inventions that were necessary for redox. I didn't actually show you this, but I was using all kinds of proteins that were metal binding, explicitly excluding only DNA regulation necessary proteins. They're not all oxidoreductases. So the fact that they all fall into the same group suggests that they may have stolen this active center from oxidoreductase proteins. And I see that cobalt and manganese and iron appear early, which is in line with what I would expect from the geological conditions. And then we think that by including the global environmental conditions, we can determine the likely timeline. We can actually put time on there rather than saying this is close to this or closer to this, and so on and so forth. So that's it. This is the people that do the work, usually. And this is the people who pay for it. So thanks. Mm-hmm. So I have been putting off looking at the individual classes until I was sure that the metric that I have was somehow meaningfully representative of time. It was not straightforward. I was hoping that I would see time, but it wasn't straightforward to me that I would. Because we're not using sequence alignments, and this is complicated. So what I did see is that the majority of the proteins in the collection are oxidoreductases, majority being about 3/4. And they are favored by experimental biases towards particular subtypes of proteins. So I would have to do a lot of debiasing that way in order to actually look at evolution. But it's also important, my suspicion is that I will not be able to see the individual functionalities because we're looking at the ligand binding center as opposed to the overall protein. And I think that that is actually transferred throughout history significantly more so than everything else. So I don't know. I think that for me, for this method, they would be instantaneously contemporary, right? So, yeah. Mm-hmm. It's, that's a very cool question. So I started looking at the sequence to structure relationship much before I started looking at the evolution of redox. So I guess during my PhD, we had extracted a subset of SCOP database where we actually had different structures, same function, same structure, different function, same sequence, different structure, and so on and so forth. And they're all very interesting cases. You can write 5 papers, and about every single case doesn't do anything in terms of overall patterns. So either we don't have enough information about outliers, which makes sense. We don't have information about outliers by definition. Or there is some freak accidents, that some environmental shifts that require appearance of new things, and they do. So great idea, no answer for you.
So I have got a question. I'm not sure how I'm going to articulate this. So it's clear that there are certain functions that have to be highly conserved.
Right.
And in fact, there are going to be certain structural conformations because those functions are so sensitive to the protein holding that that's going to be conserved as well. So if we look at proteins from modern organisms and we look at their structures, if we're looking at a very ancient function, how likely is it that a protein we see in a modern microbe is very similar to the one that was there 4 billion years ago, or even if it's conserved its function and its basic structure, will it have evolved to this, so it's only 20% or 10% conserved, even in a case where the function has to be conserved?
Right. So there are 2 answers to that. So the first part, how likely is it to be like the ancestral one? And actually quite likely. So the idea that you have something that works, it gives you this evolutionary advantage that is huge. Perhaps over changing time, things that adopt you are the accessories. So if you recall, I had this huge protein there and the center of it. So there are people who believe that metals were useful for bringing proteins together. They served as glue because it's very hard for amino acids to interact randomly. So if you bind the protein on a metal on one end, and you bind the metal on the other end, and you now have a structure that's metal binding that actually allows for protein to exist. So that's an interesting view. I don't know if I share it, but you definitely need a particular way of grabbing the ion in order to be able to do all the activities that you want to. So my guess is structurally they are very similar, even if we have to really define down to fewer residues to identify that structure. Right, so that's the answer 1. Then with the more detailed sort of explanation and the answer 2, how did it adapt? So what we have is another project that's going in the lab, which differentiates the amino acids in the protein structure as being toggles, rheostats, or neutrals. And they are hopefully descriptive of what we expect. So neutrals are just there. So you can put any amino acid in there. Nothing is going to happen to function. They're there for overall maintenance of solubility of the protein or something like that, right? Then there are the toggles, which are the catalytic site, for instance, residues, where if you remove this catalytic site, there is no activity, and that's it. So you never see it in nature when you've removed it. In fact, the threshold that you would need to pass to compensate for that removal is so high that potentially it never happened, right? And then there is the fuel for evolution position, the rheostats. And the rheostats you can keep mutating, and actually there is the majority of them are rheostats, right? So you can keep mutating these positions at length while you maintain the same architecture of the protein. So sequence identity can go down way below 10% on a regular sequence level, and even in the structure level, it could be very small. But you know, even as you saw with the Sahle score, you would have to have a really long alignment in order to have a very low sequence identity. But in the end, it's okay. It happens, right? So the structure will be maintained as long as it's functional. And functional is actually also a weird one. Most people don't think about this, but if you think about the function of any given dehydrogenase in yeast versus, I don't know, human, right? They're very different. They're doing the same, they're making the same reaction. It's just that they're doing it at very different speeds and rates. So yeah, it's the same function, but different.
So just as a follow-up, then how close are we to perhaps being able to postulate here's a set of 50 or 100 most primitive genes that you could put together and say this is sort of what that
Organism.
looked like?
It depends. Are you Craig Venter? So he can postulate right now. I don't know if I believe it. That's my problem, right? So we can postulate. We can say this is likely the minimal organism, but the reality is that we don't know what the weather was like, right? So I don't know. Really just no idea. I don't know what happened there, but no idea. I think we can come up. This was my PhD defense question. Seriously, this was a question that I was asked. My thesis was on the effect of mutations on protein function. And I had David Shaw in the room, who was on my thesis committee. And one of my advisors, not even looking at me, looking at David, said, if you had unlimited funding, how would you go about creating an organism where you can test your predictions? And the idea was if you have a minimal organism, any mutation that disrupts protein function is going to kill that organism. Right? So that was a high throughput way of testing whether your predictor works or not, if you had unlimited funding. But the problem is, of course, that you can create minimal organisms 99 million ways, and they will work. So mm-hmm. Generations. Yeah.
Try to get back to where it started. But that was never the case. They always said, oh, okay, well, we'll start evolving and everything around this sort of struck me as not a question at all.
No, but this is
amazingly cool stuff.
This is exactly, thank you. This is exactly follow-up here, that you can create a minimal organism a million ways, right? So, and then you can put it into a real life cell and it will do something else with it. Well, so the interesting part, so bacteria are a really good model organisms because they have such a short generation span and they don't have the mixing that comes with eukaryotic sex, right? But if you look at humans as a very interesting idea that most people fail to see, if you sequence the entire population of the world, you will notice that there are no mutations, right, in particular genes. And this is because mutations in these genes are lethal, embryonically lethal. So what we observe in diseases, right, is this idea that there are some not so important genes that we can mutate and get something that we as humans call disease, right? Now that's a much lower, it's a step down from let's kill everything that has that mutation, right? So that most people, okay, it's a severe disease, there is a mutation. No, severe disease was when the person didn't make it out of the embryo stage, right? So that was severe disease. And then when you define disease is also complex, right? So what a bacterium defines as disease is complicated, right? What's a disease to a bacterium? So hairless cats, is that a disease? I mean, would be a disease in humans. So what does a bacteria define as disease? And that is also important. So I think to them, it's just a matter of how many of us are out there in a particular environment. How many copies can we make? And you can take it a step further and you can say, how many copies of my genes do we have out there? And then horizontal gene transfer becomes a big deal, and ability to optimize functionality of a particular gene becomes a big deal.
So Yana will be here for the rest of today and also tomorrow morning, meeting down at the data science boot camp. We're down for the speakers zone. So if you would like to spend more time and interact with us, please let me know. And let's thank you.
Thanks. Thank you.